A very good day, my dear postgraduate students. After a long lull, we are again going to waddle through the recent advances. Be careful with the topic endocrine pathology of the lung, and let us see what all we are going to traverse. Step by step, avoid overstepping. I would like you people to kindly be very slow and deliberate. These are the things, and you shall be seeing that there can be a normal physiology, the development, the pathology of it, in spite of the usual inclusions of the pulmonary tumors, their differentiation and their own. What could be the manifestations of the carcinoid syndrome, and how do you classify the tumors, the differential diagnosis, and ultimately the references and the summary. So, I would like you people to kindly be slow and deliberate. When we talk about the endocrine pathology of the lung, there are two systems basically. One, the pulmonary nervous system, and two, the pulmonary endocrine system. You find that these endocrine cells are scattered throughout the body during the development, and they are in a linear fashion and isolated. Sometimes they can be gathering in groups, sometimes called as tumorlets. So, our aim will be to summarize the entire stretch of this endocrine pathology. See, for example, starting from way back in 1949 up to 2023, almost the latest. These are the landmarks. I would like you people to slowly and deliberately go through and appreciate the various stages of it. This I shall be coming to repeatedly. And this particular chart of it can be used for your college seminars. And this is the literature behind it. Again, you people can go through it one by one. Now, there are many physiological and pathological conditions, and you find that it starts from the neurological system down to the GAT and so on. And in this particular diagram, I have boxed in. There is the function of the pulmonary neuroendocrine cells in the physiological and the pathological conditions of the lung. For example, an encephalitis is there, and then there is a vascular change which can lead to a lung injury, which we usually see in viral infections. Again, there can be a pneumonia or a bronchial asthma, whatsoever. So all of these lead to a panel of reactions such as a muscular contraction and otherwise. So there are some critical sensors which we shall be seeing hopefully. The pulmonary endocrine system in the normal lungs, the structure and development. So it is found in all the species. This is supposed to be one of the most primitive species, the bashir and the squid. So the gill epithelium, wherever there is an epithelium, it is said that the endocrine shall and will be there. It can be in solitary cells in case of non-innervated organisms and in clusters in case of innervated. And they are usually located at points of branching. And in the adults, they are more organized, clustered, innervated, integrated neuroepithelial bodies. In fact, the word carcinoid itself means organoid, which is more like an endocrine system. So the endocrine differentiation occurs from the neoplasm, and you find that there is a very deft difference between the epithelium and the endocrine. So much so that both can be simultaneously occurring in certain conditions. There are various secretory products. The importance of it is this. It is not that, as in the case of IHC, that the same tumor is going to produce the same product with the same effect. You find it can vary from species to species and also from person to person. Serotonin, gastrin-related peptide. This is extremely important. Calcitonin, others. All these may be produced to varying extents individually or in groups. The neuroendocrine products, again, you find that these are the cells we are talking about. There is a respiratory membrane, calcitonin, and the other products are all there. Type 1 and the type 2 pneumocytes. And finally, you find there can be some expressor cells that are also being stimulated by the brain, leading to the Alzheimer's, Parkinson, inflammation, and death. So there is a subtle connection between the neuron, endocrine, and the pulmonary systems. The functions of the pulmonary endocrine system is not very clear, at least then. This article is quite old. Neuroepithelial cells are mainly concerned with hypoxia. One key word that I would like you people to remember. The paracrine regulation of the pulmonary growth also. So the pulmonary growth depends on the endocrine system, the development. And finally, after an injury, the restoration of the pulmonary tissue. And these could be identified by means of various antibodies to the antigenic markers, such as neuron-specific kinase, PGP 9.5, and chromogranin, antigens in varying endocrine cells. So this is an important table for us. As I said earlier, it is not limited to tumors. So I am finding a lot of disease conditions in the lung which are non-neoplastic. Take, for example, I have in fact shortlisted this particular table. Asthma, pneumonia, bronchiectasis, cystic fibrosis, emphysema, and pulmonary tumors, eosinophilic granuloma. Then ventilation and oxygenation, tumor-led, sudden infant death, and brainstem injury. So all of these are in a way directly or indirectly connected with the endocrine system in the lung. The patterns of proliferation. So you find that these are the things, there is a lot of diversity that is available in the end stage. There is a linear cell distribution. Sometimes it can become nodular, as I had mentioned earlier. And these are the various products which can be produced by it. Gastrin-releasing peptide, very important. ACTH, vasoactive intestinal peptide, somatostatin, growth hormone, and alpha HCG, not beta HCG. And see what is happening over here. Depending on the oxygenation and the carbon dioxide, you find that, as in the case of a bronchial asthma, etc., there can be a vasoconstriction, then a bronchoconstriction, various other things which are again happening by means of the liberation of various substances. So this is true. There is a linear endocrine proliferation. The arrow marks here are supposed to be, probably I'm able to see a little organized one. These are inflammatory cells which are being scattered, but in the subepithelial region, there is a linear endocrine proliferation that is seen. And in some cases, they can become nodular, called as tumorlets. These are inflammatory or tumor-like lesions, and they resemble the carcinoid tumors, hardly measuring about 2 to 3 millimeters in diameter. Tumorlet by itself can be a question for you. This is again another diagram. I had taken it from ResearchGate. Look at this. There is the epithelium over here. Maybe there are some goblet cells. There are some secretory cells. And then the ciliated epithelium. All of these are developing from a basal cell. So the basal cell differentiates into whatever cell that is required. And what happens is there can be a proliferation again. So depending on the need and the irritation or whatever it is, you find that there can be a horizontal and then a vertical proliferation like this. Sometimes what happens, there is a stimulation for a particular one. The columnar epithelium might be replaced by a squamous epithelium and it proliferates. These can also be because of genetic alterations. And you find that there are further genetic alterations. Ultimately, you find that there can be a normal tissue proliferation and a glandular one, or there can be some features of cellular atypia, anaplasia, etc. So thanks to ResearchGate for this beautiful one. So progressively you find that there is a stem cell which is undifferentiated, and then there is a differentiated one which can again become neoplastic. So this is somewhat similar to your cervical intraepithelial neoplasm. See what is over here. Diffuse, idiopathic, pulmonary, neuroendocrine cell hyperplasia, DIPNIC. I do not know whether this is a question for us. Horrifying questions we all get. So this is one. It is almost bland. And then you find that there can be a conversion into the typical carcinoid with less mitosis. And it is more organized and regular. Then it can become atypical. Look at the variation, the hyperchromaticity, and so on. Sometimes there can be areas of necrosis, increase in mitosis. The atypical carcinoid. And then there can be large neuroendocrine carcinoma, as well as the small endocrine carcinoma. So these are the things. So small lung cell carcinoma, large lung cell carcinoma are also supposed to be overlapping on this. That we shall be having in mind. The explanation is given over here, and thanks to the reference. Types of pulmonary neuroendocrine tumors. This is the same thing. The typical, atypical, large neuroendocrine, and small cell carcinoma. And what are all the features? The grading, the morphology, mitotic index, and the necrosis. Thanks to thoracic. Manifestations again. You find that morphologically there can be four major things. Dense core vesicles can be developed in these. There can be other endocrine markers. Enzymes can be produced, as well as secretory products. So these are the various things. For example, the vesicles can be seen under histochemistry or direct observation, argyrophilia. So that is the lead hematoxylin, immunolabeling, and chromogranin, etc. can be used to study these. Other endocrine markers, this will be the batch of markers that can be used. S100 protein, for example, classical of any neurological lesion, and synaptophysin again. And enzymes such as dopa carboxylase, diamine oxidase, creatine kinase, and neuron-specific kinase. So all these again somewhat related to the nervous system. And the secretory products are given over here. This particular one you need not memorize, but please do have an idea about the spectrum of occurrence as well as the development of these things. And this is being reflected in this. What we are seeing over here is I'm finding large cells which are somewhat atypical, and here they are all small cells. There is overcrowding of the cells over here, and here again there is another one. So these all will have to be studied morphologically, immunohistochemically, and at the molecular level, courtesy this particular reference. Pattern. Always a carcinoid is an organoid, and an organoid is supposed to be an endocrine. So I hope you people can recollect an organ or a tissue like the pituitary or the parathyroid, wherein you find nests of cells which are more or less uniform, and you need not very keenly see. If I see one part of it, you need not see the other parts. It is a duplication. That is exactly what we see in any carcinoid tumors, and so also in the lung neuroendocrine. So it is a low grade, and probably the size is also small. No necrosis, mitotic figures are low, thanks to this reference. Now, in the earlier classification, there are very simple ones. That is what is called as a dysplasia, carcinoma in situ. Finally, there is a squamous cell carcinoma, adenomatous hyperplasia, and again carcinoma in situ, adenocarcinoma, and the diffuse idiopathic DIPNIC, and the neuroendocrine carcinoma. These are the three major things. It is too simplistic for the postgraduate student to write. Therefore, the WHO has given this, which has been periodically getting renewed. So this is what is there. The epithelial tumors, you find that the entire list is there. I am not going to read it. You people are in fact better than me. So there is a lung neuroendocrine neoplasm. Ectopic tissue can be the mesenchymal hematopoietic. Usually I say this as a mnemonic. So epithelial, non-epithelial, miscellaneous, secondary tumors, unclassified tumors, and tumor-like lesion. ENMSUT. Absolutely meaningless, but that has helped me. So molecular imaging again. Here I find that there is a PET scan that has been taken to identify it. Look at this, very well circumscribed. It is not as irregular as a bronchogenic carcinoma. And these are the pairs of lungs that I am seeing. And in this one, look at the mitosis, arrow marks, courtesy atypical carcinoid lung pathology outlines. And this again, typical and the atypical. Here it is somewhat what I had explained earlier, the nests and the trabecular or the insular patterns that are over there, and they are all anastomosing, almost uniform. Whereas here, they are overlapping, there is a crowding. Sometimes you find that there can be a smashing of the nucleus, all can be there, along with areas of necrosis, atypical pulmonary carcinoma. This one is extremely important. Now, there are two things. I would like you to concentrate on the left side first. The lung neuroendocrine neoplasm 2021 classification. So it is the lung neuroendocrine neoplasms, of which you have got one as dip neck, which you have already seen. Diffuse idiopathic neuroendocrine hyperplasia, given for the sake of completion. That is what the author says. But then there is a well differentiated from which you can have a typical and an atypical lung neuroendocrine tumor. Also, there can be a lung neuroendocrine tumor directly, which can be morphologically having the features of a well differentiated large cell, small cell, as well as there can be a combination of the neuroendocrine carcinoma and the lung tumors. Again, what is important in this will be there are a series of this. If you go through the earlier classifications, I've just taken out the keywords. They have been progressively called as carcinoid, then arising from the Kulczycki cells, atypical carcinoid, intermediate squamous or small cell carcinoma. There can be a combined one, and non-small carcinoma with neuroendocrine differentiation. These are all the various terminologies, and that has finally come over here. So I would like you to see this one. Endocrine differentiation in other pulmonary tumors. There are three pairs and the panel of antibodies which have managed to put in a box. Two from the dense core vesicles, chromogranin A and LEU7. Two secretory products, GRP and ACTH. Two reliable markers for endocrine, PGP 9.5 and synaptophysin. This alone we shall memorize. This is another important one. What are all the features? Whenever we talk about the features, always think of the subheadings. The subheadings will take you to the answer. So the major types here are carcinoid, atypical carcinoid, large cell carcinoma, small cell carcinoma, and there can be an overlap. This we all know. Though these are the subheadings. Just like your bone marrow, here also you have a set of subheadings. Architecture, growth, stroma, necrosis, cell, cytoplasm, nucleus, chromatin, nucleoli, mitosis, N/C ratio. And in this orange one, I have highlighted the salient point of it. For example, you find architecture is organized in the case of carcinoid, others have just left. Growth is very regular, carcinoid. Stroma is conspicuous, it becomes less progressive. Necrosis widespread in small cell carcinoma. And you find that the cytoplasm is scant, increased in NC ratio. And the shape of the nucleus almost all oval to round to fusiform. Chromatin, you find that there can be a dispersed or a stippled chromatin. It can be coarse in a case of large cell. Mitosis is increased, and you find that NC ratio is also increased. Please concentrate only on the orange background boxes. There are various secretory products. This I am not going to repeat again. And there is something called a closed loop manner. Probably it is something like the thyroid that we are seeing. So there is a hormone that is being liberated, it acts on, and then after that there is a negative feedback. A similar such thing is being happening. So there is a kind of regulation which is happening. And thanks to WebPath, this is again a tumor. In this, I am able to see some kind of an orderliness. There is a spindling of the nucleus. There are, of course, mitotic figures. It can go in for a metastasis. Endocrine neoplasm, there are various parts. As I told you, there can be nest-like cells, there can be a glandular pattern, there can be rosettes, there can be, again, an insular pattern, etc. And there are various enzymes which can be produced that I have mentioned. So there has to be a coordination between radiology, microscopy, and immunohistochemistry. For example, this is INSM1, thanks to Cleveland Clinic for this particular picture in the identification of this particular thing. And here again, by the PET, I'm able to see a well-circumscribed lesion within the lung. And look at the various patterns of it. I'm seeing islands and islets of cells. There are anastomosing cords over here. There are some kind of rosettes and pseudo-rosettes which are being formed. Again, isolated linear forms which are there. Thanks to ResearchGate for the patterns. As a conclusion, we see that it is important to find the role of the pulmonary endocrine in the healthy lungs. So it has been there and it persists till our death. Probably there will be some amount of decrease and a destruction with the various inflammatory lesions. They form the pathogenesis of some pulmonary diseases. Overt endocrine differentiation can also occur, and there is an established one. And influence on the behavior and the treatment are now under being studied, and this is there. So some of the references, the others I have already quoted in the text of it. So assuming the radiology and then the histology, what else are we to remember? You find that this will be the list of subheadings. Think of the subheadings, it will take us to the answer. Thank you.